Welcome back to the journal club that's for the classics papers in immunology. This particular paper is from 1979. We went up to 1980 last week. This week we're going back a little bit to 1979. I didn't want to shift the emphasis too much on everybody, on the audience. And so if you really like this video and the journal club in a general sense, don't forget to click on the YouTube video that you liked it. Click on the bell so you get notified for more. Also scroll down on the YouTube site. You'll see all my links to my website and so forth. And go on my website and click on the writing tab and then scroll way down. You'll see all the classic papers are there. So you don't even have to go to the library in these days of the pandemic, and that's good. The paper that we're going to talk about today is the authors are Steve Gillis, who is my first graduate student, Gerald Crabtree, who was a pathology resident at Dartmouth, and he decided to take a year off and do some research with Alan Monk, who was one of my really good collaborators, and I'll tell you about him in a minute. And the title of the paper is Glucocorticoid-Induced Inhibition of T-cell growth factor production, the effect on mitogen-induced lymphocyte proliferation, published in the Journal of Immunology in 1979. Just to give you a little background on these glucocorticoid studies. When I came back to Dartmouth after spending a year as a postdoc in France, I became an assistant professor for the first time. So I sort of finally came of age. I wasn't a postdoc anymore. It turns out right upstairs from our laboratories in the Remsen Hall of the Dartmouth building was a fellow by the name of Alan Monk. And Alan was a famous endocrinology researcher. And his background is that he graduated, he got a PhD in, I think it was chemical engineering. He was an engineer, really, which made him a little different. He thought in equations rather than words. You know, he's one of those kinds of people. He did a postdoc at MIT after he did his graduate work. And the postdoc that he did was in a laboratory that was studying hormones. And Alan, after he left his postdoc, while he was at Dartmouth, he published the first paper describing glucocorticoid hormone receptors. Now, glucocorticoids are hormones that are made by the adrenal gland. It sits on top of the kidney. And the connection with immunology is that it turns out that there are glucocorticoid receptors, according to Alan, in every tissue in the body that he looked at. One of the most sensitive tissues that they found were the lymphoid tissues, and in particular the thymus. And he found that if you would administer to a mouse or a rat a big dose of glucocorticoid hormones, the thymus would involute. It would just shrink away within 24, 48 hours. And so that was his target organ, and thymocytes were his target cells. And that's how he established his glucocorticoid assay. Now, since I had been trained as a hematologist, I knew that glucocorticoids, they were really one of the only immunosuppressive drugs available at that time that you could write a prescription and get it from the pharmacy. And they were not only immunosuppressive to people that had allergic diseases or autoimmune diseases or any inflammatory disease, they worked very well. But in addition, glucocorticoids were used as one of the primary treatments for lymphocytic leukemias and lymphomas. And again, they would shrink down the leukemia. It was quite impressive. And nobody really knew how the glucocorticoid hormones regulated these lymphocytes. Alan had done a lot of experiments on studying the metabolism of thymocytes primarily, with and without glucocorticoids and different doses of glucocorticoids. And he found that the metabolism of the cell, and in particular, he devised an assay for the uptake of glucose from the milieu, from the medium that he had it in, was inhibited by glucocorticoids. And so that's the background. And when I started up at Dartmouth, I started talking to him, and he was a very good guy. I really liked him. And so he convinced me that we should be looking at glucocorticoid receptors in human cells, because we might be able to do some experiments to figure out why they were so effective in suppressing lymphocytes. the introduction of this paper. So this is published in the Journal of Immunology, and in the introduction, we point out straight off that the effect of glucocorticoids on lymphocytes in vitro was really first published in 1961 by Peter Noel, who, as you may recall, first published on the idea his finding that there were mitogens that would activate lymphocytes to start to proliferate, become large blastoid cells and undergo mitosis and proliferate. And that paper, the first paper, was published in 1960, and then the very first experiments that Noel did thereafter was that he put glucocorticoids into his cultures, and he found that it inhibited, which wasn't surprising because people knew that they were inhibitory to the immune system. And he also found, and he published in that paper in 1961, that he had to have the glucocorticoids in there early. If he activated the lymphocytes with phytohemagglutinin and then waited a day or two, and then added the glucocorticoids, the suppressive effect on the mitotic index was markedly attenuated. So it had to be there early. And the going notion at the time, or dogma, was that the lymphocytes in humans, small resting lymphocytes, were glucocorticoid resistant. But then after you activated them, they went through a sensitive glucocorticoid where you could inhibit them, and then if they got beyond that phase and underwent mitosis, they would become resistant again. That was sort of to explain this whole phenomenon. And so that's what Alan and I started working on together, and it resulted in my first Nature paper, where we showed that when you activated the cells with PHA, the first thing we did was measure the glucocorticoid receptor number, because that's what we knew how to do. And so we found that the receptor number went up 2 or 3-fold when you activated the cells. So that was sort of strange because it was paradoxical. We thought maybe they would go down to account for this physiology of this whole thing. And we also then measured metabolic assays that Alan had already established in the laboratory. And we found that both the resting lymphocytes, as well as the activated blastoid lymphocytes, the glucocorticoids would depress the metabolism of the cells, glucose uptake and protein synthesis and other things that we measured. So then the question was, why were glucocorticoids so inhibitory? They didn't kill the cells. They were not cytotoxic, which was one of the things that a lot of people thought at the time because of their effects on leukemia and lymphoma. So then, as time went on, so we published that, and as time went on, my lab became focused on the proliferative response of lymphocytes, as you know. And we discovered that there was this mysterious T cell growth factor that seemed to be responsible for the proliferative response of the cells when you activated them either with PHA or concanavalin A mitogens, or when you activated them with specific antigens. It's the same kind of thing. So once we realized that what we had in this kind of thing was a whole new system, and we could assay for the ability of the cells to produce the T cell growth factor, and separately, we could also assay for the ability of the T cell growth factor that we could add to actually cause the cells to proliferate independently of one another. So that was what we had started out to do in this series of experiments that was published in this paper in 1979. So in the materials and methods, the growth of our assay cells, for the TCGF assay cells, the ctll, cytotoxic T lymphocyte lines, we kept them in culture, as I've said in some of the other episodes of our journal club, at a low cell density. We would seed them at like 10,000 cells per ml, let them grow up to about 100,000, and then we would split them down again, and we split them basically every day. So we always had cell cultures in the incubator that we just split, ones that had been split yesterday and the one that had been split the day before yesterday. And we kept those cells. Our source of our growth factor was concanavalin A activated rat spleen cells that we culture for 48 hours. And we used about a 50-50 mixture of our conditioned media and fresh media. Now, for the glucocorticoid assay, this is the way that Alan had devised it. And in these experiments that we did in this paper, Jerry Crabtree is the one that was performing the receptor assay, because that's what he had been doing in Alan's lab. So in this instance, this assay was performed in these tiny microfuge tubes that are about 1 ml. And they're really handy because you don't want to have a lot of liquid and the whole thing. And you can manipulate them in these, what they call a microfuge, so that we have microtubes and microfuges. And so we had 2 million cells per each tube. We would then expose those cells to a dexamethasone. A dexamethasone is a synthetic glucocorticoid, and it's one of the most potent of glucocorticoid hormones. And we could buy it at the store already radiolabeled with tritium, which gives off beta radiation. And so we would put these cells in these multiple tubes in dilutions of the glucocorticoid, and we would incubate them for only 20 minutes, because the glucocorticoids would go right through the plasma cell membrane and find its glucocorticoid receptor in the cytoplasm. And that all came to steady state within 20 minutes. And Alan had all worked out all this stuff at 37 degrees. And he also taught us that this is how you do physiological assays, because he was in the Department of Physiology. So then we would stop the reaction by cooling it rapidly to 3 degrees, putting them on an ice slurry in an ice bucket, and then spinning them in the microfuge at 5,000 times G for 15 minutes. And we then take off just 20 microliters of the soup and put them in separate tubes to count for CPMs, counts per minute. And that would allow us to calculate, knowing the specific activity of the hormone, the concentration of the hormone in each tube. And that would be the free concentration outside the cells. And then we would dilute the whole thing 60-fold in 1.5 nanomolar magnesium chloride. And that Alan had worked out so that the cell would 1.5 nanomolar is basically water. And so that the cell would puff up in like a fat Albert and pop. But the magnesium chloride would allow the nucleus to stay together. And so then you we'd vortex it vigorously at that stage, and then let it sit at 15 minutes at 3 degrees, and then centrifuge it this time at 12,000 times G, pelleting all the nuclei. And then we would take off the supernatant and count the nuclei in the counter, and that would be the amount of bound hormone. And so you only had 2 things to measure: free, bound and free. And then we would plot the data by the method of Scatcherd, which had been worked out in 1949. And you can plot it other ways, but you can plot it sort of on the y-axis is bound, and on the x-axis is free, and you'll get a saturation curve like that. Or you can plot the ratio of bound over free on the y-axis against the molecules bound on the x-axis, and you get a straight line curves if you only have one affinity of a receptor. And where it crosses the x-axis is a direct readout of the number of receptor sites per cell. So that's slick, and that was good. So that's how you do the glucocorticoid receptor assay. We measured then in my laboratory, and Steve and I would measure the glucocorticoid effect on the ctll cells themselves. So we would put them into culture, and either in microtiter plates, and then let them grow for a while at varying concentrations of dexamethasone, and then we would pulse them with tritiated thymidine and see whether or not the dexamethasone had changed that. And we could also put them in larger cultures and then count the cells in the tissue culture plates every day, so we can get cell counts and so forth. The TCGF assay that we used to assay for whether or not the dexamethasone or the glucocorticoid would influence that was the one that we reviewed a couple of weeks ago. And we used, as I went through in that journal club, we went through the probit analysis and the way you plot the data to get straight line plots that gives, then you can get a very direct readout of the number of the concentration of TCGF expressed in units per milliliter. And so we then devised several other assays using either rat spleen cells activated with CON-A or mouse spleen cells or human peripheral blood lymphocytes activated with PHA, and then harvest the supernate plus minus dexamethasone, harvest the supernate to see whether or not the glucocorticoids influence the amount of TCGF that cells could make. So in the results, we found that when we looked at the number of glucocorticoid receptors per cell, because we hadn't measured that in our ctll cells, so we wanted to make sure that at least they had glucocorticoid receptors, and they did. They had about 5,000 or 6,000 sites per cell, and glucocorticoids would bind to them with a characteristic dose response curve so that the equilibrium dissociation constant could be calculated from this, I forgot to say, the slope of that line that comes down and crosses the bound molecules per cell. And the slope gives you the equilibrium dissociation constant, or the KD. We did a bunch of experiments showing that the assaying for glucocorticoid, true glucocorticoid hormones, and also looking at the binding of other kinds of steroid hormones like estrogen and testosterone and so forth. And we found that the binding was glucocorticoid specific. So now we need to do some screen sharing. So once we had assayed for glucocorticoid receptors in the ctll cells, the next thing we wanted to do was to see whether or not there was any influence of glucocorticoids on the growth of the ctll cells. And in table 1, we've got data from a single experiment, but this was done many times, where we cultured the cells either for 24 hours or for 48 hours. And then in this column is the dexamethasone concentration. And because dexamethasone was dissolved in ethanol, this is the ethanol control of no dex. And then this is 10 to the minus 6th molar all the way up to 10 to the minus, or down to 10 to the minus 10th molar. And you can see that there was a diminution of the tritiated thymidine incorporation from 91,000 counts per minute. And if you just put that as a percent of control over here, and then at 10 to the minus 6, the highest dex concentration brought it down to 65,000 counts per minute, or a 30% diminution. And then 10 to the minus 7th was 86, but by 10 to the minus 9th, or 10 to the minus 8th, it was only a 90% reduction and so forth. And the same general kinds of data that we got at 48 hours. So that was tritiated thymidine. It wasn't too impressive. And just a little bit of a, not enough to explain the rapid involution of lymphocytes in these in vivo experiments that Alan had done. And so then we went on to counting the cells. Here we have growth curves of the ctll viable cells per mil, and these are the days of culture. And you can see that, let me just get, this is culture medium alone in the triangles. And you can, this is a typical growth curve of ctll cells growing over 5 days. And then at 10 to the minus 10th and so forth, down to, or up to 10 to the minus 6 molar here. Again, it's a diminution, but it's not, 100%. So then we said, well, let's see whether or not there's an effect of Dex on TCGF production. And so we did the experiment, and this is with rat spleen cells activated with concanavalin A. So we have two things listed here. In the open bars are the is tritiated thymidine incorporation of the cells, and we cultured these cells for 48 hours. And then in the crosshatched bars is the amount of TCGF produced, taken from the supernatant and assayed in the TCGF assay. And so you can see that when there's no con A added, there is no thymidine incorporation. And if you look at the con A, the ethanol control for the dexamethasone, there is a lot of thymidine incorporation and a lot of TCGF produced. And then at the highest concentration of dexamethasone, 10 to the minus 6 molar, and then as we dilute it out, we lose the effect. But you can see that there's a marked effect of dexamethasone on the production of TCGF by these cells. The effect on these cells was glucocorticoid specific in the inhibition of TCGF production that's shown in table 3. Dexamethasone inhibits the ethanol control down here is 0.88 units produced, and prednisolone, which is a very active glucocorticoid, also inhibited. Cortexolone did not inhibit. Cortisol did inhibit. And then we have deoxycorticosterone, which does not do it. Corticosterone does it to a certain extent. Progesterone, estradiol, testosterone, cortisone. Cortisone has to be activated in vivo. None of those worked. And so it was a glucocorticoid specific effect, which is what we were looking for. We could show the same thing. We found the same thing with rat splenocytes and also with human PBMCs. Here's an experiment that's with human PBMCs, and it's exactly the same sort of phenomenon. So it was pretty clear that what was going on is that there was an effect on the proliferation of the cells when you assayed for when you gave them T cell growth factor. But if you asked the cells to make T cell growth factor, they really couldn't do it. The last experiment that you really wanted to find out was well, if you added T cell growth factor to the cells in the presence of a dilution curve of dexamethasone, these are mouse spleen cells, these are rat spleen cells, and these are human cells. You can see that in the crosshatched bars is when we're adding the TCGF, and they're proliferating just fine. And so the effect of the glucocorticoids were basically very, very distinct. So that's it. The TCGF overcomes the DEX10 inhibition of the proliferation. And in the discussion of this paper, we said, okay, there's a dual mechanism going on here. There's a little bit of inhibition by the glucocorticoids on the ability of the cells to proliferate in response to TCGF, but it's minimal by comparison to the effect on TCGF production. And in addition, there is no cytotoxicity. The cells, we did the cell counts and so forth, and there was no cytotoxicity. And the glucocorticoids lengthened the doubling times of cells, but they did not kill the cells. So we speculated in the discussion of this paper that maybe maybe the total lymphoid cell mass in the host, in the animal or the human being, was being regulated by glucocorticoids, because the experiments had shown that if you adrenalectomized an animal, a mouse or a rat, then the thymus would grow in size and the lymph nodes would get bigger. And so there seemed to be a sort of a governorship on the lymphocytes and the whole lymphocyte compartment by glucocorticoids. And I'm not aware that whole mechanism has been pinned down ever since these experiments were done and reported. But we'll get into some of these things about what the thymus is all about later. The other thing we pointed out in the discussion of this paper was that our experiments and our results really explain Knoll's data from 1961, from, I was 20 years previously. And that is that the mitotic effect of the glucocorticoids it was inhibited by the glucocorticoids, but that it had to be present early on. And now it's obvious why, because the T cell growth factor is produced within the first 24 to 48 hours. And we also pointed out that this also is consistent with the findings of Billingham and Sir Peter Medawar from 1951, when they showed that skin graft rejection could be delayed if you treated with glucocorticoids, treated the mice with glucocorticoids, but you had to pre-treat glucocorticoids. If you waited 48 hours after placing the skin graft, then you would not get a delay in the rejection. The rejection would proceed just fine. And of course, that's important nowadays in 2021, from 51 to 21. So that's what, 60 years. With COVID, pneumonia that they get is not due to the virus. Well, it's indirectly due to the virus. The virus comes in, gets into the lungs. It's the immune response to the virus and the immune response of the production of large amounts of cytokines, IL-2, in addition to other interleukins, as well as the pro-inflammatory cytokines, as we'll get into. That causes the capillaries to leak in the lungs and the lungs to start to fill up with fluid. If you give glucocorticoids to those patients, you have to give it as early as possible. And that's probably what happened to our wonderful President Trump. Swifted him off to the hospital and gave him they threw the kitchen sink at him, essentially. But one of them was, I think the major influence were the glucocorticoids. So that brings us up to the present time. The impact of our paper from 1979. The other thing that we talked about in the first paper is that, well, if this is true, then and particularly as a takeoff on Medawar's experiments, that means the generation of cytotoxic T cells, the creation of the cytotoxic T cells that you could do in vitro with a mixed lymphocyte cultures, that probably is inhibited as well by the glucocorticoids, and then that's why, and you have to give it early on. So the accompanying paper, the number 2, is in this series, is the effect on in vitro generation of subtle cytotoxic T cells, and basically the glucocorticoids inhibit the generation of cytotoxic T cells. Now, and the other thing that happened then is soon thereafter, so we published this and we told people about it, and this is when I first started talking about hormones to the general field of immunology, and because I had been essentially educated by Alan Monk as to what hormones are, how you define them, and how they work in the body and so forth. And I've already had that because I had been educated in medical school rather than graduate school. So endocrinology was, it was one of the courses I had to take. And so I knew all about hormones and hormone receptors and that sort of thing. And so I started saying that these lymphokines are really hormones, it seems to me, and they're hooked into the other hormonal systems, the adrenal gland and the adrenal hormones and so forth. And not only that, they are probably also involved in the neurotransmitter hormones of the central nervous system. So there's all kinds of connections going on, and it's a very complicated system. I don't think that the idea of hormonal regulation of the immune system and the immune response has ever been really understood and accepted by the immunologic community. And we'll go into that in spades as we move forward. There was a paper that came out soon after we published these two papers with a similar story with regard to Cyclosporine A. And that was published in the European Journal of Immunology by Hermann Wagner and Martin Rollinghoff from Mainz, Germany. And they basically showed that Cyclosporine A, which was a new drug, it had not been introduced in the clinic yet, but Cyclosporine A in combination with glucocorticoids really supplied the recipe for blocking graft rejection. And soon thereafter, and basically what Wagner and Rollinghoff showed in their EGI paper is that it blocked TCGF production. If you added TCGF yourself, you could overcome that block. And so it was a very similar kind of mechanism. As time went on, in the 80s and then into the 90s, the molecular mechanisms of how steroids, glucocorticoids, inhibited TCGF production, as well as Cyclosporine A, and they were similar in terms of their molecular mechanism of inhibition, but they were different. So that meant that you could use them in the clinics together and they would synergize, and you could lower the dose, and which would lower the side effects of each individual drug and still get a maximum effect. And after that, there was another drug that was introduced from Japan that was called FK506. And that also blocked TCGF production, was found to block. And then after that, there was another drug that was introduced called Rapamycin that was isolated from a culture of bacteria that had been taken from Easter Island in the middle of the Pacific Ocean, and which is called Rapa Nui is the name of the island, or Easter Island. And rapamycin was found to block not the production of TCGF, but the activity of TCGF. And so you could combine all of these drugs because they all were suppressive, but they worked by a little bit different mechanism. And that really was the birth, essentially, in the 80s of the whole field of organ transplantation. Up until these drugs came along and we understood their mechanism of action in terms of inhibiting TCGF production, there was only really one drug for immunosuppression, and that was the glucocorticoids. And I don't know if you know the ancient history of organ transplantation, but Christian Barnard from Cape Town, South Africa, we hit the newspapers all over the world with the first cardiac transplant in 1968. Only problem was that he could hook up the vessels and take the heart out of a cadaver and put it into a live person and hook it all up, and it would still work. And so this was hailed as just a revolution in medicine. The only problem is the immune system rejected that heart, and that fellow only lived a few weeks after the heart transplant because they didn't have really good immunosuppressive therapy. These drugs that emanated from our initial finding with the glucocorticoids put the surgeons into the operating room, and now we basically transplant lungs and pancreas and all kinds of organs, even faces and so forth and so on. That's the beginning of the idea that what you can manipulate the immune system and the immune response by manipulating these cytokines and these lymphokines, which gave impetus to going forward and finding out more ways that we could bring the laboratory findings into the clinic. So I'll stop there, and don't forget to like the video if you like it, and click on the bell and check out the other papers that we're now into the early 1980s, and things are going to start to get hot, and I think you'll like it. So I'll stop there. Thanks.